Now, when we enlarge this interaction, and what we're going to see is that there's a nerve ending, there's the muscle cell itself, but they're not really in touch. They're not directly touching one another. Now, we didn't know this when we could use light microscopes, because the light microscope couldn't give us that detail. But when we went to an electron microscope, then what we saw was that there is no direct contact between the nerve and a muscle cell. So we have this space, and so what we need is a compound that will move across the space to create a connection. So last week we were saying those were what we call neurotransmitters. And then the neurotransmitter has to have a receptor that it binds with, okay? And so that's why we need neurotransmitters, because neurons really aren't touching other entities, all right? So let's look at that in detail. So what we're seeing here is we're seeing the motor nerve ending coming down and associating with a muscle cell. So now what we can do is we can add some detail to this whole process. So a space is a cleft. The interaction between a nerve and another nerve or a tissue is a synapsis. So the space that exists in the interaction is called a synaptic cleft. So the word is descriptive, synapsis meaning the interaction between a nerve and something, and then cleft meaning is space. All right. So what the nerve has to do is it has to make a neurotransmitter. So we can divide neurotransmitters into protein-like and fat-like compounds. So if they're protein-like compounds, you have a gene on a DNA. The gene is transcribed to messenger RNA. It goes out into the cytoplasm. Rough ER manufactures it, and the Golgi apparatus packages it. So what we see in the synaptic end bulb, which is the end of the nerve, are the synaptic vesicles that contain our neurotransmitters. So for skeletal muscle, the neurotransmitter is acetylcholine. And those are pre-made, pre-existing. So that we have these synaptic vesicles of acetylcholine in the synaptic end bulb. So what's going to happen is when you stimulate a nerve, and so it's the brain that's going to initiate this process, create a muscle contraction, then the impulse is going to be carried down the nerve to the synapsis. And when the nerve impulse arrives at the synapsis, it's going to cause these vesicles to migrate to the surface and release the neurotransmitter. Now, we're going to describe in detail how that works when we do the nervous system. So right now, you just need to know that when the impulse arrives, it causes the secretory vesicles, the synaptic vesicles to migrate. Okay? And then what we're going to do is we're going to call the position of where this occurs, the membrane on the cell is going to be called the motor end plate. So this area is referred to as a motor end plate right here, where the nerve is going to interact with the stuff, with the neuron. So it's our motor end plate. And when we look at the membrane of the motor end plate, then we have specialized things that acetylcholine is going to bind with. So that when acetylcholine is released, then it's going to bind at this receptor. So this is called a receptor. Okay? And then once it binds with the receptor here, then it causes the receptor to change what it's doing. Okay? And then if we want to be able to communicate, we cannot have acetylcholine lasting forever. So when acetylcholine is released, it is quickly broken down by an enzyme that breaks down acetylcholine into the acetyl group and the choline group. That way you don't release acetylcholine and have it last for hours, because then you wouldn't be able to control what's going on in your body. So you need acetylcholine to be released and broken down quickly, so that if you need to have it released again, you have to stimulate the release again. Okay? So what we're going to see is that within a membrane, we create a difference from the inside of the membrane to the outside of it. Okay. So if it's inside the cell, then typically we would say cytoplasm, right? But what if we wanted to specifically say that we're dealing with a muscle cell? Then we could get rid of cyto and do sarcoplasm. And then we know that we're talking about the cytoplasm within a muscle cell, right? Now, what do we call the fluid around cells? Intracellular fluid. And what about, we kind of used a different word, so it was IF, interstitial fluid, yeah. So even though you look at a muscle, and the muscle looks like everything's in contact with each other, we are a bag of water that's providing an aqueous environment for our cells. That's why we have that keratinized stratified squamous. So we can create a pond ecosystem for our cells. And if we don't do that, then what happens is all our tissue shrivels up. So what muscle tissue would look like if you couldn't retain the water would be what you take out of a bag of jerky when you go to the grocery store, which is just dehydrated muscle tissue. Yeah. So to prevent ourselves from becoming jerky, we keep ourselves moist. So what we do then is, anytime we have two compartments, then we have a membrane. And so our membranes considered freely permeable or selectively permeable? They're selectively permeable, which means that not everything can pass through, right? So what we can do is we can maintain concentration gradients across the membrane. So what we do in our interstitial fluid is have a lot more sodium. And there's an ion that likes to hang around with sodium all the time, which is chloride, negative ion. And we have a lot less potassium outside of our cells. So inside of our cells, we have less sodium. We have more potassium. We have phosphorus as a negative ion. Plus we have cellular proteins. That also tend to have a negative charge to it. So what we keep is a concentration of positive and negative ions separated across the membrane itself.
Sorry, I didn't catch you. On the interstitial fluid, you've got the sodium, you've got
chloride.
Fluoride?
Chloride.
Chloride.
Cl is chloride. Like in table salt, sodium chloride. Right? And then less potassium. Okay. So because we maintain concentrations across the membrane that are different, then we can actually create a charge disparity across the membrane. So on muscles, what we end up with is, because of the dominance of sodium outside, we have a positive charge, negative, relative positive charge to the inside of the membrane. And then because of the phosphate plus these negative cellular proteins, then we end up with a slight negative charge on the inside of the membrane. So that we get a charge disparity across the membrane. And that's the way we can actually now carry current, because we actually have this charge disparity across the membrane. So what we end up with then is that when a membrane is rusting, we have a charge that we can actually measure, which would be minus 70 millivolts. The difference between the inside and the outside of the membrane. Okay. And if the membrane isn't doing anything, we say the membrane's at rest. Now there's one other piece of the puzzle here, and that is that membranes are, if we elevate a membrane, membranes, remember, are bilipid layers. So that we end up with a polar part of the membrane to the outside. The center of the membrane is nonpolar. And so we end up with polar toward the sarcoplasm, polar toward the IF, and nonpolar in between. So since we have that nonpolar layer, that's why our membrane becomes selectively permeable. So it's going to be based upon the size of the molecule, the charge of the molecule, on what can move through a membrane. Okay? So what happens then is that certainly sodium will move through a membrane, potassium will move through a membrane. If you look at a periodic table, sodium is a smaller molecule, potassium is a larger molecule, or a larger ion. So what happens is sodium can go inside the cell to some extent, and potassium can leak out of the cell to some extent. So to maintain that, what we have is an active transport mechanism that's constantly moving ions in and out of the cell. So remember, if we're moving two entities together, in the first unit we called it a co-transport system. And when we looked at co-transport systems, we could divide them as symporters or antiporters. So which one moves ions in opposite direction? An antiporter. Yeah. So what we essentially have in skeletal muscle is an antiporter system. And what it's going to do is it's going to move 3 sodium outside of the cell for every 2 potassium that it moves inside the cell, because sodium runs through at a faster rate. So what we typically do is we just call this thing a sodium-potassium pump. And as long as you have energy, because this is active transport, it requires us to use ATP. As long as you have energy, you can maintain these ion imbalances. And you have to maintain the ion imbalances to make this whole system work. So not surprisingly, if you run out of energy, you decide you're not going to eat ever again, then you're eventually going to stop working because you don't have the energy to run these things like this anti-porter system, which we have to run to make it work, okay? So, now we're ready to go on here. So what we're going to do is, if we look at our motor end plate down here, is this thing allows sodium to pass into the cell. So what really happens is acetylcholine or acetylcholine is going to bind with the receptor. It actually opens up what we call a sodium channel. And that's going to allow sodium to go from the interstitial fluid, which is out here, to the sarcoplasm, which is in here. Okay? It's going to allow sodium to go from the interstitial fluid, which is out here, to the sarcoplasm, which is in here. Okay? So our receptor is actually what we call a channel protein. And since it allows sodium to move, we say it is a sodium channel. And then what we can do with a sodium channel is we can open it or close it. Okay. So before acetylcholine binds with it, it is closed. And if it's closed, sodium can't pass through. And if we unite acetylcholine to it, then it opens and allows sodium to move into the cell. So we're using acetylcholine to take and transfer an electric event from a neuron that's technically not in contact with the nerve, with the muscle, to the muscle. and creating that electric event in the muscle itself.